Okay, so previously I talked about how to download a PDB file for a protein from RCSB PDB. Right now I'm going to discuss about what information can we have from this page that opens up when you click on any ID. Okay, so from here that says download files, we downloaded the PDB format file. You can even download the FASTA sequence for that protein and any other format file according to your use, according to your need. Then this is the 3FGN is the PDB ID for this protein, the crystal structure of D-thiabaltin synthetase. Each protein has a unique ID in the PDB, right? For example, if this particular protein is having a complex with any enzyme, coenzyme, any cofactor, or it is complex with any other protein, it is going to be having a different unique ID. Then this is the method that was used to determine the crystal structure of this protein. It says that the X-ray diffraction method was used. The resolution is 1.85 angstrom. This is the 3D report on the structure validation. Then this here, the macromolecule content, it tells you about the weight of the macromolecule in the kilodaltons. Then it lets you know about the total number of atoms that make up the macromolecule. This protein is made up of 6,648 atoms, right? Then you can have an idea of the total number of amino acids, that is 907. Then it says that this protein is having a unique chain. Unique chain, it means that it is having only one possible sequence for the set of sequence for the chain, right? Literature box, this tells you about the literature that has been published on this protein. You can even search it on the PubMed, you can download it from here. Then this is much detailed view about the protein. It says that it is having 4 chains, and earlier we observed that this protein is having one unique protein chain, right? So it means that these 4 chains here, they are having the identical sequence. It means that this protein is active in its tetramer form, and one chain has a length of 251 amino acids. This is the experimental data and validation, the same thing I discussed earlier. Okay, so as in my previous video, I selected the entry with no bound ligand or compound. It shows no small molecule or micromolecules, right? So in order to determine the information of the complex amino acids and the coenzymes, let's say you have clicked onto the protein ID that is having the small molecules attached to it. So this is how the detailed information comes. It says the any other compound that is attached to the protein along with its 2D diagram, the 3D interactions. You can click here and you can observe what were the interactions between the ligand and the protein. crystal structure of mycobacterium and tuberculosis dithiobiotin synthetase complexed with dithiobiotin and phosphate. See, this is the detailed view of the interaction between the compound protein, or you can say the interaction in the protein complex. This bond shows the hydrogen bond. Okay, so you can actually put the cursor right on the this dotted line, and you can determine the type of the bonding in between the ligand and the protein's active pocket, right? So this is the importance of the ligand or the compounds that are attached with the protein in order to determine the activity of the protein's active pocket. This could later help in our study to determine the nature of the compounds that might be present at the active site, so that is determining the type of interaction in between the ligand and the protein's active site. So for now, let's just keep it till here, and in the upcoming videos I will be talking about on how to visualize the protein on camera and how to create an interactive chimera diagram of the protein. And please don't forget to like and subscribe my channel, my content, and do share it if you find it worthwhile. And yep, thank you.